Hi, welcome to my Bachelor final project presentation. I'm Martina Serrat, and I'm going to introduce you my work on neuroscience from a gender perspective, supervised by Olga Valverde. It was Darwin himself who said, "The chief distinction in the intellectual powers of the two sexes is shown by man attaining to a higher eminence in wherever he take up than woman can attain." Such words can be surprising, especially coming from one of the most relevant figures in science. However, it evinces that neuroscience from its origins has historically co-evolved with the social perception of the moment. Inversely, discoveries belonging to sex and gender have a strong power into society. That's why we should ask ourselves, how is the gender question addressed in investigation? Before we start, we can revise some terminology. Contrary to used terms indistinctly, sexual features are refer to those genetic, gonadal, and genital attributes which are traditionally seen as internally consistent and discrete. Whereas gender entails a set of symbolic and sociocultural characteristics related to behavior patterns, expressions, and identity of girls, women, boy, men, and gender diverse people. With that in mind, the overall aim of this project is to serve as a state-of-the-art review of sex and gender brain research by analyzing the knowledge production procedure, its behavioral and structural findings, but also the causes underlying these observations. To finally set a basis for the development of new strategies in science, but more precisely in neurobiology. Therefore, we are going to analyze the research process involved in this field from the very start till the publication and future implications of findings. The first step on project design is determining the assessed variables and consequently define the methodology. When looking for simplification and malleability, animals, well, essentially rodents and non-human primates, imply the main source of human biological data inference. Interestingly, the first anatomical discovery on sex research was found in songbirds, which present distinct singing patterns and nuclei. However, direct translation of data from animals to humans might be compromised. Plus, only sex variable is accessible due to their poor socialization. While in human studies, data accessibility relies on indirect neuroimaging techniques or postmortem samples, sex assessment is limited to the studies of early development. After the first minimum socialization, sex and gender are indistinguishable in terms of cognition. Therefore, covariate and any potential external influences should be acknowledged in the studies. In past, clinical studies were based on rare cases, such as hormonal dysregulation, like congenital adrenal hyperplasia. Nowadays, research opts for more longitudinal studies with larger samples of diverse individuals, controlling, for example, their hormonal levels. Most human studies, however, show a sampling bias as well. The numbers in participants are usually low for statistical significance and misrepresenting different collectives, being caused maybe by volunteering bias or an oversight by specialists. It is crucial for data robustness to analyze the observations accurately. When looking for potential differences, effect sizes, which means magnitude, and confidence intervals rather than statistical significant differences and standard error should be regarded. More precisely, psychological or behavioral observations focus on verbal, visuospatial, and mathematical abilities, and also stress coping system or toy preference are narrowed and rather fluctuating from modest to negligible in magnitude, depending on the assessed psychological domain. As for anatomical and functional observations, multiple areas are under investigation. However, I'll explain just a small set among the most commonly assessed structures. Verbal abilities can be structurally inferred as well, through the planum temporal. As for hemispheric connectivity, apart from connectome studies, corpus callosum has been analyzed, seeking for an explanation for females' supposed multitasking abilities. These statements have been refuted through meta-analysis, finding no significant differences in temporal symmetry and callosum shape or volume. Other telencephalic regions arise special interest, such as the hippocampus and amygdala, being crucial for memory and spatial abilities and presenting differences in shape and cellular aspects, such as dendritic spines. Although when adjusting the structure volume for the total or intracranial volume, these differences vanish. Furthermore, one of the most characterized areas in sex and gender studies is the preoptic area, embracing two antagonic subnuclei, the intermediate nucleus and interventral periventricular nucleus. Despite potentially representing reproductive functions, observation in these areas overlap between sexes, which means that at least one-third of the population shares a similar volume, far from their respective male or female average. Finally, a nearby region, the bed nucleus of the stria terminalis, and another interstitial nucleus of the anterior hypothalamus, the third, have been examined as well, plus claim to be related to gender identity. Although most of the reporting studies lack of sample size reliability and gender representability, like this study from Garcia and Falgueras, which only represents one individual, trans female, and doesn't even use the correct term. On the whole, up-to-date findings must be thoroughly inspected for their potential bias. Thus, considering observations as dimorphic or differential in sex is questioned by the evident overlap between groups. Some authors even argue that in such cases, measures are not predictive of sex or gender. Opposite to that, there is an effect of sex. It is also claimed that female or male typical structures can coexist within a single brain, a theory which is called brain mosaicism. Now, on a further extent, experimental communication is likely to be subjective and consequently biased as well. Due to journal editorial practices favoring positive results, studies with negative or null results, which are the equivalent to similarities or lack of differences for sex studies, are subject to the phenomena called file drawer problem or time lag bias, which means that there is a tendency to underreport these results by authors or underpublish them by editors and keep them in a file drawer. Regardless of the outcome, special care should be taken when defining the nature of observations and to discuss result reproducibility. The multiple and intricate factors influencing sex but also gender must be regarded. On a first level, in early fetal development, chromosomic component of fetuses determine gonad differentiation to ovaries or testes. The resultant hormone expression completes the genitalia development and shapes over other tissues, including brain. Hormone fluctuates over time and context and has been proven to influence it on brain in the traditional organization and activations processes in early periods, but also in adulthood. Similarly, environmental factors vary as well, depending on social situation, time, place, social groups, and self-perception, and work as a potential influences. All these factors converge in an epigenetic pattern modification, either DNA methylation or histone modification. The resultant up or down regulated genes shape brain structures in a transient manner by promoting or inhibiting neurogenesis, cell death, and survival, neuronal migration, and morphological changes in any lifetime moment. So, although there is a strong influence from genes and hormones, socialization and self-perception can also shape behavior and brain. Empirical differences should be interpreted as a result of intricate dynamic interactions of social and biological processes, evidencing the need for a social approach in sex and gender brain research. The current review had a quite ambitious goal entailing multiple and distinct issues on neuroscience. Therefore, hampering the development of a specific and concrete analysis. However, it gives a broader perspective in these terms, contributing in this open discussion to enhance the development of gender and health research. Hope you enjoyed this presentation and thank you for your attention.